Abnormal regulation of gene expression can lead to developmental abnormalities and cancer. As we saw in Module 1, cancer results from genetic changes that affect cell cycle control. There are two classes of genes associated with cancer: those that stop or slow the cell cycle and those that trigger cell growth and division by initiating specific phases of the cell cycle. Tumour suppressor genes repress the cell cycle, initiate DNA repair or control cellular adhesion. An example is P53, known as the guardian of the genome. Mutated P53 gene is seen in a number of cancers. Any mutation that decreases the normal activity or expression of a tumour suppressor gene can contribute to the onset of cancer. Suppression of the cell cycle is important if the cell's DNA is damaged. The release of protein P53 suppresses the cell cycle and prevents the cell from passing on mutations to new daughter cells. Here, prolonged exposure to UV light causes DNA damage in a cell. In a normal cell, the damaged DNA triggers an internal signal that is passed via protein kinases, leading to the activation of P53. Once activated, P53 gene is transcribed and translated to P53 protein, which is a transcription factor. The P53 protein binds directly to DNA, arresting the process of cell division until repairs are made. Mutations in P53 can inhibit expression, so the inhibitory protein is not synthesized and the cell cycle is not inhibited. Mutated cells divide uncontrollably, leading to cancer. While tumour suppressor genes suppress the cell cycle and therefore growth and division, proto-oncogenes code for proteins that promote cell growth and division. Proto-oncogenes include genes for growth factors, their receptors, and the intracellular molecules of signalling pathways that control the cell cycle. These genes are tightly regulated to prevent uncontrollable cell division. Defects in the regulation of proto-oncogenes cause these genes to stimulate growth by triggering the cell cycle. Different types of genetic changes cause proto-oncogenes to turn into oncogenes. These may be chromosomal translocations, where the gene is under different control, or gene amplification, where excess protein can be made, or point mutations causing excess or hyperactivity of the mutant protein. The RAS proto-oncogene, when mutated, converts to RAS oncogene that can lead to increased cell division. RAS acts as a messenger between a growth factor receptor on the plasma membrane and a cascade of protein kinases. In a normal cell, cell division is triggered when the growth factor binds to its receptor in the plasma membrane. RAS passes the signal to a series of protein kinases that activate transcription factors, which then activate genes to produce proteins that stimulate the cell cycle. This normal pathway of cell cycle stimulation through RAS will not operate unless triggered by an appropriate growth factor. However, mutations in the RAS gene can lead to the production of a hyperactive RAS protein that stimulates a protein kinase cascade, even in the absence of growth factors, resulting in increased cell division and potentially cancer. More than one mutation is needed to produce all the changes characteristic of a fully fledged cancer cell. The incidence of cancer increases greatly with age because mutations that occur throughout life accumulate and the likelihood of developing cancer increases. At the genetic level, the multi-step model of cancer development means that a cancerous cell is usually characterized by at least one active oncogene and the mutation of several tumour suppressor genes. Colorectal cancer is one of the best understood types of human cancers that fits the multi-step model. Nearly 13,000 new cases of colorectal cancer are diagnosed each year in Australia and New Zealand, and it usually develops gradually. The first sign of this cancer is the development of a polyp, which is a small benign growth of cells in the colon lining. Polyps usually develop due to mutations in a tumour suppressor gene such as APC. The cells of a polyp, although they look normal, divide frequently. As the polyp grows, it can become malignant due to gradual accumulation of mutations that convert proto-oncogenes into oncogenes. such as activation of RAS genes and by knocking out p53 tumour suppressor gene. Individuals can inherit oncogenes or mutant alleles of tumour suppressor genes, and as a result, these people are more susceptible to colon cancer. Mutations in the BRCA1 or BRCA2 genes cause an increased risk of breast cancer. The Australian Cancer Research Foundation report that a woman who inherits one mutant BRCA1 or BRCA2 allele has a 60 to 70% lifetime risk of developing breast cancer compared to the general population risk of around 10%. Around 5 to 10% of breast cancers occur in women whose families have a BRCA gene mutation. Tar derivatives that are associated with cigarette smoking are known to be mutagenic and carcinogenic. Lung cancer from smoking probably involves 2 or more genes. This graph shows the risk of developing cancer by the age of stopping smoking. As you can see, the risk of developing lung cancer increases along with age, but the relationship isn't linear, suggesting that the longer an individual smokes, there is an increased risk of accumulating mutations in various genes, and therefore a greater risk of developing lung cancer. This suggests that there's more than one gene involved. Now let's reflect on the learning objectives we've covered. We should now be able to describe the 2 main types of genes involved in cancer, which are tumour suppressor genes and oncogenes, and how these genes are involved. And give an example of each. Explain the multi-step model of cancer development that we saw in colorectal cancer. And you should be able to explain what type of cancer is associated with mutations in the BRCA1 and BRCA2 genes, and how these genes impact cancer risk for individuals with mutations in these genes. Thank you for watching.